Okay, let's get started. Welcome everybody to today's QAAFI Science Seminar. I am Mike Gidley, and it's been my privilege to be one of the advisors for today's speaker. So, a great honor to be able to introduce Xianyi Liu, who's coming to the end now of his PhD journey, and will tell us all about what he's been doing. So, the next slide, please. Before we get going, let's acknowledge, on behalf of the University of Queensland, the traditional owners and their custodianship of the lands on which we meet, and pay our respects to ancestors and descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. So today's seminar is scheduled to be from now until at the latest 1 p.m. Please feel free to put questions into the Q&A tab, not the chat function. So I won't read the chat function, but I will read the Q&A tab. And then after the presentation, I'll come back and facilitate the questions to the presenter. So today's presenter is Xianyi Liu. He's in the final stages of his PhD candidature and has been doing a project that spans not only disciplines, but also academic and commercial outcomes. So from this point of view, he's a good example of what a QAAFI PhD student should be doing, great science for great outcomes. And without further ado, I will get Xianyi to introduce the topic that he's going to talk about. Over to you, Xianyi.
Okay, thanks, Mike. Just double check. Can you guys hear me? Okay, wonderful. I also acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respect to their ancestors and their descendants. So let's get started. Good morning, everyone. I hope you are doing well. My name is Xianyi Liu. I am a final year PhD student at CNAFS here in QAAFI. So thank you for taking your time to join this science seminar today. What I will be presenting today is a summary of some of the major incomes from my PhD project. And as you can see from the title here, my project is about optimizing feed digestibility in pigs using enzyme supplementation. Just to briefly introduce the background of this PhD project, my PhD project is part of a big ARC linkage project titled How to Make Antimicrobials in Pig Feed Redundant Naturally. This ARC project basically contains 3 programs, each program focusing on a different aspect of the project. The first program, which is led by Professor Eugenie Rora, focuses on the maternal conditioning of piglets, as well as promoting piglets' robustness during the post-weaning period. My colleagues, who are also PhD students, Sally Taylor and Paul Bulgari, are working tirelessly on these 2 projects, and they have got some really promising findings. The second program, which is the one I'm involved in, as you can see, is about optimizing feed digestibility using enzymes. This program is led by Mike, who is also my principal supervisor, and what I will be talking today are also the findings within this program too. The third program, which is led by Dr. Conitoni, as well as my other colleague Lenny, focuses on improving piglets' microbiome and gut health through nutritional strategies. So together, these 3 programs will produce a collaborative outcome, which is a feeding program that can promote early intake of a highly digestible diet to support post-weaning piglets. Before I start, I would like to acknowledge all the efforts that everyone has put into this project. My work would have been impossible without the support from my colleagues, as well as the project funding bodies and collaborators, as you can see at the bottom. So I would like to start this presentation by talking about pigs. Why do we farm pigs? We farm pigs because the final product is pork. They are food. According to the FAO, pork is currently the most consumed meat worldwide, followed by poultry. And the term pork also includes processed meats such as bacon, ham, salami, etc. Over the past 10 years, as you can see here, pork has overridden poultry to become the top consumed meat in upper middle income as well as high income countries. And it is projected to maintain its position for the next 10 years as well. In Australia, during 2024 to 25, the value of pork industry was around $7 billion. So we do produce a certain amount of pork at here. And talking about pork production, the major expense or cost within the current production context is actually feed cost. For the countries that there is some data available, you can see that a vast majority of the production cost for every kilogram of pork sold is denoted to feed cost. And in Australia, the feed cost can take up to 70% of the total production cost, but it can fluctuate much more since we have to import a lot of ingredients from overseas. So there has been a question around for years within the pork industry worldwide, which is how do we minimize feed cost to maintain the profitability of the business due to recent inflation? And there have been some approaches around, such as using alternative locally sourced ingredients or minimizing feed processing to save on energy cost. But in my point of view, any approach will have to come down to maximizing feed digestibility, simply because that every penny you invest into the feed should go into pigs rather than being excreted into the environment. So again, the principle of minimizing feed cost, in my point of view, is to maximize feed digestibility. That is to maximize the nutrients that go into the pigs and minimize the nutrients that come out of the pigs. And having good feed digestibility is not just about profit. It's also about pigs' welfare. If we can get the pigs eating well, so they can stay healthy and having a low risk of disease. So having a good feed digestibility is also about improving pigs' welfare as well as reducing the use of antimicrobials in pig industry. In pig production, pigs generally experience 3 to 4 production phases, starting as suckling piglets to weaning pigs and then growing and finally finishing pigs. Within these 3 to 4 production phases, there is one time point that is especially important for pigs, which is the weaning transition. This weaning transition in pigs is very different to what we do as humans. For us, when we have babies, we wean the babies at around 6 months old, and then we gradually giving them solid food until they can get used to them. But for pigs, this weaning process is just a one-day event, and within that one day, the pigs, you can see here, these suckling piglets, they are separated from their moms and then transported to a different housing system and mixed with other litters, other groups of piglets. And also within that one day, these piglets, they have to stop drinking their mom's milk and suddenly switch to eating a solid diet. And why is that one-day weaning a problem for feed digestibility in piglets? If we look at the digestive enzyme development in these piglets before weaning, which mostly happens at around 4 weeks' time, their digestive system is quite developed to digest milk, which contains a lot of lactose. So you can see they secrete a lot of lactase. But by the weaning time, that happens at around 4 weeks, their digestive system is not mature enough to suddenly switch to digesting a solid diet, which basically requires the action of a bunch of enzymes. So what I want to say at here is that if you look at the amount of enzymes like amylase, protease, lipase, these are the enzymes that they need to for digesting solid diet, and these enzymes will only gradually build up over the next few weeks post-weaning. And that is currently a major industry challenge, which is the poor feed digestibility right after weaning. And this problem is mostly due to the sudden change from drinking sow's milk to eating a grain-based solid diet. If we look at the composition of sow's milk, we can see that it is quite nutritious for piglets. It contains a lot of lactose and fats. It contains easily digestible proteins. It contains digestive enzymes that are originated in the milk, as well as some growth factors such as bioactive peptides. But within that one-day weaning transition, they will have to suddenly switch to digesting a grain-based solid diet that has a completely different nutrient profile. And this nutrient profile is basically formed by starch and other complex carbohydrates, complex plant proteins, there will be no enzymes and no growth factors. So a research question that we had here was that how do we make sure weaning piglets can extract the best value from solid feed in every single bite post-weaning? And to answer that question, we first need to have a good understanding of the composition of pig diets. Regardless of region, pig diets, they are mostly grain-based, with cereal grains supplying most of the starch as digestible carbohydrates, also as the major source of energy, and oilseed meals contributing to the majority of dietary proteins. So starch and proteins, these two are the largest nutrients in pig diets. In Australia, we use a lot of barley and wheat to fill up the cereal fraction of the pig diets because we grow barley and wheat here. And for the oilseed meals, we use a lot of soybean meal and canola meal. So these two oilseed meals, they are just byproducts of oil extraction from soybean seeds and canola seeds. Sometimes you might also see that pig diets have some animal protein sources like blood meal, fish meal, meat and bone meal. So those animal protein sources, they are much more expensive than these vegetable sources, and in most cases, they are just used to fill up the amino acid and mineral requirements for the pigs. So how do we improve feed digestibility? We need to know where the nutrients are in grains. In the grain crops, nutrients are mostly located in the endosperm of the grains. And if we zoom into endosperm, we can see that nutrients such as starch and proteins are mostly located inside the endosperm cells, which are encapsulated by these plant cell wall. So when we feed these grains to pigs, what happens is that during digestion, the digestive enzymes that are secreted by the pigs are mostly blocked by the cell wall outside these plant cells, making them unable to access and digest the nutrients inside the cells. So this is what we call the cell wall barrier effect. The cell wall barrier effect is actually a major cause of incomplete digestion for many nutrients in grain-based feedstuffs. So what if we use some enzymes? What if we use some non-starch polysaccharide degrading enzymes? We can potentially compromise this plant cell wall and release the nutrients for better digestibility. So this is a major way of how we can improve starch and protein digestibility as well as for many other nutrients. So apart from the cell wall barrier effect, there are two other factors that can also affect nutrient digestibility or feed digestibility, which are the phytate and the structural protein. For phytate, it is a famous anti-nutritional factor because it has the ability of binding to a lot of nutrients and making them inaccessible for the digestion by pigs. And beyond that, I also wish to point out that phytate can also affect starch digestibility because it has the ability of binding to calcium, and calcium is an important cofactor for amylases to function properly. Another factor that can affect nutrient digestibility in grains is the structural protein. In the grain endosperm cells, so you are seeing this is a barley endosperm cell. These proteins, they are bulk proteins. They are nutrients by themselves. They are also structural proteins. So structurally, you can see that these proteins, they are mostly located on the surface of starch granules, just like a coating. And this coating effect will partially restrict the excess of amylase towards these starch granules, which will then lead to reduced starch digestibility. So a hypothesis that we had here was that supplying protease can enhance starch digestibility primarily through removing or degrading this protein coating effect. So to improve feed digestibility, we know that an effective approach is to use exogenous enzymes as feed additives, and they can tackle anti-nutritional factors. Enzymes, by definition, they are biological catalysts, and they can speed up biochemistry reactions. In pig feed, we use them as feed additives to improve feed digestibility. I just wish to emphasize on the term here, exogenous. Exogenous means anything that is not from the pigs. So the enzymes that we we use in feed as feed additives. They are not from pigs. They are not produced by pigs. They are rather produced by some bacteria and fungi. So when we put enzymes into the feed, of course they're used to improve digestibility primarily by degrading the anti-nutritional factors. So down here you can see this is a very simple illustration of how enzymes work. Enzymes basically they can bind to their corresponding substrates and break down the substrates into products. So when we put enzymes into feed, what they can do basically they will bind to the substrates, and the substrates for those enzymes can be any anti-nutritional factors. They can be phytate, they can be non-starch polysaccharide, they can also be starch or protein. So that's basically how enzymes work in feed to improve digestibility. So regarding those enzymes, what has been done in pigs? And basically, a wide range of enzymes that were produced from different sources at different dosage have been given to pigs at different ages, fed with different diets. And the studies have all reported different results at the end. So instead of presenting you what study has reported what result, I will show you what they haven't done too much, which are the gaps of knowledge. We propose that there are basically three gaps of knowledge. The first gap was that the enzymes have been quite extensively studied in the animals, but they haven't been evaluated too much for their individual effects with separated feed ingredients. So how they work on individual feed ingredients. And this can pretty much only be done through in vitro methods. The second gap was that even though the enzymes have been studied quite well in the animals, we think there is still a lack of literature on how the enzymes could influence the nutrient digestibility in post-weaning piglets at different sites or different sections of the small intestine, particularly during the first week post-weaning. And the third gap, which was built on the second one, was that the enzymes, the commercial enzymes that we explicitly studied in my PhD project, they have not been comprehensively evaluated for their effects on the apparent digestibility of different nutrients in pigs. So the aim of this project was to understand the effect of enzymes on the nutrient digestibility in pig feeds with a focus on post-weaning piglets. And according to those three gaps of knowledge, we designed three studies which basically studied enzyme effects following a process from in vitro to in vivo, as you can see. The first study, which was an in vitro study, this study focused on testing the enzyme effects in individual feed ingredients. And then as directed by the results from this first study, we moved on to testing the enzymes in vivo in piglets 1 week post-weaning. And the third study, which was the last study, we tested the enzymes in ileocannulated pigs to study their effects on the ileodigestibility of nutrients. So I will now go through the studies one by one. The first study, which again was the in vitro study, was to evaluate the effects of different exogenous enzymes, different enzyme additives on the starch digestibility of individual grains and to study their mechanisms of action through an in vitro model. The results from this study have been published on this paper. And if you have any interest, please feel free to look it up online. So in this experiment, we tested basically 4 enzymes against 3 hammer-milled grains, which are hammer-milled barley, wheat, and maize. And we studied their effects on the starch digestion of these 3 grains using an in vitro digestion model. This in vitro digestion model was based on InfoGest 2.0, and to measure the starch digestion, we basically used 2 parameters, which are the amount of starch digested and the digestion rate coefficient, each representing how much and how fast starch was digested. The in vitro digestion model that we used was based on InfoGest 2.0. This digestion model basically simulates digestion in 3 phases, including an oral mixing phase by mixing the grains with feed enzymes, a gastric digestion phase with or by porcine pepsin, and a small intestinal digestion phase by porcine pancreatin. And during the small intestinal digestion phase, we took digester out at different time points to analyze for the two starch digestion parameters. So here are some results. The first result that I would like to share with you is that the feed enzymes improve the starch digestibility of the 3 grains to different extents. For example, in barley and wheat, you can see that xylanase gluconase, which is the red color down here, and kumbol, the orange color, they showed really good effects in improving the amount of starch digested at the end of digestion. However, for maize, you can see that quite different. Only phytase and kumbol significantly, well, you cannot tell the significance for now, but they showed pretty good effects in maize. So again, this is the differential effects that you can see on this page. And apart from visualizing the data, we can also analyze them using statistics. So on this slide, I'm just using the red colors to highlight the enzymes that significantly increased the 2 starch digestion parameters, or maybe one of them, compared to control. So for example, in barley, you can see that xylanase gluconase and kumbol significantly increased both the digestion rate coefficient and the amount of starch digested compared to control, while phytase also significantly increased the digestion rate coefficient. In wheat, similar effect was seen by the xylanase gluconase as well as kumbol, while phytase and protease significantly increased the amount of starch digested. In maize, however, only phytase and kumbol significantly increased the 2 starch digestion parameters. So again, what I wish to emphasize at here is that what you see at here, the differential improvement by the enzymes, these are the major results that we found within this study. The second result that I would like to share with you is some microscopic evidence supporting the effect of xylanase gluconase on the grain cells. So what we did, we basically took some digester out of the small intestinal digestion, and then we analyzed them under microscope, a light microscope. So this is what we have seen for barley, and I think this contrast is quite clear. For the barley in the control group, we have seen a lot of undigested starch granules that are still encapsulated by the barley cell wall, even after 8 hours of small intestinal digestion. But by using some xylanase gluconase, we've still seen the cells, but the starch granules have disappeared. They have been either digested or released. So we think this observation has actually aligned with our hypothesis earlier that xylanase gluconase can effectively compromise the plant cell wall and release the nutrients for better digestibility. Similar effect was seen in wheat as well as in maize. So for maize, now we know that this cell wall barrier effect is not really a rate limiting factor because we put in the xylanase gluconase and we've seen the starch granules have been released, they have disappeared, but we did not really see a sharp increase or sharp improvement by the xylanase gluconase. So we started wondering what was really limiting the starch digestibility in maize, or more specifically, how does phytase improve the starch digestibility? So to answer that question, we designed another set of study. We basically did in vitro digestion of maize without and with phytase, and then we monitored the pancreatic amylase activity change during the in vitro small intestinal digestion. So from the results, we can tell that overall, the pancreatic amylase activity reduced during the 6 hours of small intestinal digestion, regardless of phytase. But by supplying some phytase into maize, pancreatic amylase retained significantly higher activity compared to control, which is the one without phytase. So we think this observation has proposed a potential mechanism of how phytase can improve starch digestibility, which is by preserving more pancreatic amylase activity during the small intestinal digestion. So here are the conclusions from the first study. We found that xylanase gluconase and kumbol improved the starch digestibility in barley and wheat, while phytase and kumbol promoted starch digestion in maize. We've also found some microscopic evidence supporting the use of solanase glucanase, and at the end, we proposed a potential mechanism for how phytase can improve starch digestibility, which is primarily through preserving more pancreatic amylase activity. So, as directed by those in vitro results, we move on to testing the same enzyme combinations in vivo in piglets 1 week post-weaning. The objective of this study was to understand the effects of protease, solanase glucanase, and their combination on the nutrient digestibility in 2 diets in post-weaning piglets. This study basically followed a 4x2 factorial design, testing the effects of 4 enzyme treatments, including control, protease, solanase glucanase, and their combination in 2 diets, which were both based in wheat and barley, but containing either soybean meal or canola meal as the major protein source. All the diets were isonitrogenous, isoamino acidic, and isoenergetic, and phytase was included in all the treatments, in case you wondered where we put phytase. In this experiment, we used a total of 48 male piglets with 6 replicates per treatment across the 8 treatments. All the piglets were individually housed throughout the 9 days experimental period. The piglets, they were weaned at 21 day old, and then we first gave them 8 days of adaptation period with ad libitum access to one of the 8 diets and water. On the evening of day 8, we fasted them for overnight, and then from the morning of day 9, we restrictively fed a quarter of their day 8 feed intake for every 2 hours until they were euthanized later that day. For the sample collection, we took their small intestine out, and we divided their small intestine into 5 sections, including duodenum, proximal middle distal jejunum, as well as ileum, and then we took digester out from each of the section. For the digester samples, they were analyzed for several nutrient digestibility, but today I will just present to you the starch and protein, which are the 2 major nutrients in pig diets. These are the variables in this experiment, and based on these variables, we designed this statistical model, which is basically to answer 3 questions. And these questions are, how do enzymes affect nutrient digestibility in pigs in post-weaning piglets? How do protein sources affect the nutrient digestibility in post-weaning piglets? And do enzymes improve the nutrient digestibility differently in diets with different protein sources, as you can see here? So how much digester we've got from the post-weaning piglets? On this slide, you are seeing the amount of digester that we retrieved from each small intestinal section as dry weight, gram per pig. So at here, you can basically see that we did not really get anything from the duodenum. We got a little bit of stuff, around 1.5 gram from the proximal jejunum, and most of the digester was actually concentrated in the last 3 sections of the small intestine. So most of our analysis was actually performed in the digester from these 3 sections because we got enough digester, but for the proximal jejunum, we still managed to do a starch digestibility with that 1 point something gram. So how much starch was digested by the post-weaning piglets in their proximal jejunum? As you can see, overall, the statistics tells us that enzymes and protein sources did not show any significant effect on the starch digestibility, even though you can see there is quite substantial numerical increase by the enzymes. And this lack of statistical significance, we think, is due to the insufficient number of replicates. But again, I just wish to elaborate on this a bit more. These numerical improvements are quite amazing. Protein sources had no effect, which is not surprising, and the enzymes did not perform differently in diets with different protein sources. In the middle jejunum, things became more interesting. Enzymes had a significant effect on the starch digestibility with the solanase glucanase, significantly increased the starch digestibility from 29% in control to 48%. So that was 19% increase. Protein sources did not have any effect on the starch digestibility, and the enzymes did not perform differently in diets with different protein sources. For the protein digestibility, enzymes and protein sources did not show any significant effects. And again, even though you can see some numerical increases, and there was no interaction between enzymes and protein sources. In the distal jejunum, you can see that enzymes had a significant effect on the starch digestibility, with all the enzymes significantly increased the starch digestibility compared to control. Protein sources, once again, had no effect on starch digestibility, and there was no interaction. For protein digestibility, which is different this time, enzymes had a significant effect, with the xylanase glucanase and the protease combination significantly increased the protein digestibility by 12%. Protein sources did not have any significant effect. There was a borderline effect, and enzymes did not perform differently in diets with different protein sources. And finally, in the ileum, again, enzymes had a significant effect on the starch digestibility from 90% to 95%. And in terms of improvement, you can see this is quite interesting. The protease showed very similar improvement to xylanase glucanase as well as to their combination. So all the enzymes improved the starch digestibility to the same extent. Protein sources had no effect on the starch digestibility, and there was no interaction. For the protein digestibility, enzymes had a significant effect again, with the combination significantly increased the protein digestibility compared to the control as well as the protease group. Protein sources had no effect, and there was no interaction. So for now, you have seen these digestibility measurements at each small intestinal section, and they are just single data points for each section. What I then did, I just plotted them out. I plotted the starch and protein digestibility out to visualize these digestibility progression. And that here, for example, in the starch digestibility, what you can see that is that in terms of the extent of improvement or the numerical increase, you can see that the enzymes showed much more effects in the 3 jejunum sections compared to the ileum. So the gaps between the enzymes and the control are much bigger than in the ileum. And same trend can be observed in the protein digestibility here. Even though we are missing the proximal jejunum data, enzymes actually showed much more effects in the jejunum sections. So for me, this progression or changes in the extent of improvement by enzymes means that there might be a digestion rate difference. So enzymes have done much more in the jejunum sections than in the ileum. And we think there might be something much more interesting in the jejunum beyond the 1-point digestibility measurements for every section. So here are the conclusions. Within this study, we found that protease, xylanase glucanase, and their combination significantly improved the starch and protein digestibility in the piglets fed with diets that already containing phytates. We've also found that protein sources do not have any significant impact on the starch and protein digestibility, and enzymes do not perform differently in diets with different protein sources. The third study, which was the last study in my PhD project, this study was conducted late last year, and we are still analyzing some of the results from this study. But I included this study into the presentation today because I wanted to show you what we have seen for now. In this study, we basically did an ileal cannulation, and it was to study the effect of the enzymes on the ileal digestibility of pigs. So you might see here that the term I use is a bit different. In this study, we had to use weaning growing pigs, which are a bit older than the post-weaning piglets, because there is a minimum surgery requirement, a minimum age requirement for the ileal cannulation surgery to be done. For that surgery, I will talk about a bit more in the next slide. The design of this study, basically it followed a 3x2 factorial design, testing the effect of 3 enzyme treatments, including control NSPAs and NSPAs plus protease in 2 diets, which are both based in wheat, but containing either soybean meal or canola meal as the major protein source. All the diets were isonitrogenous, isoamino acidic, and isoenergetic, and phytase was included in all the diets. So this design is very similar to what we have used in the previous study, apart from some minor adjustments in the enzymes as well as the diets. So why did we do ileal cannulation? Because ileal cannulation is considered as a gold standard for studying feed digestibility in pigs. And ileal cannulation, just like what it literally means, we put a cannula into the ileum of the pigs. And what that cannula does is that once pigs ingest some feed, the digester will flow through ileum, and some of the digester can come through this cannula, so we can collect them for analyzing nutrient digestibility. And because this cannula is inserted at the terminal ileum, whatever comes out of the cannula pretty much represents what nutrients have been digested. On the left, you can see this is a ileal cannulated pig with a cannula attached to it, and there is a plastic bag attached to the cannula. So that's how we collect digester examples from this cannulation system. In this experiment, instead of taking digester samples out from each section, we put a cannula at the end of the ileum, at the terminal ileum, and then we took digester out through this cannulation system. So in this study, we used a total of 12 ileal cannulated pigs with 6 replicates per diet across the 2 diets. And then within the same diet, the 6 pigs were randomly assigned to one of the 3 treatments and then rotated between the 3 treatments for 1 week per treatment. So for example, if one pig was assigned to control, then it will be on control for 1 week and then move to the next treatment for another week and the last treatment for another week. So together, this is a 3 weeks experiment. And also within these 3 weeks, we do not do digester collection all the times. We took digester out only for 2 days. So for 2 days, we will open up the cannula and attach the plastic back to it. And during this time, we fed them 3 times of the maintenance energy to make sure that they get enough nutrients to meet the nutrient requirements. So I'll quickly finish this due to the time limit. For the apparent ileal digestibility of starch, we found that enzymes and protein sources both had significant effects, again, on the starch digestibility. So you can see here that in terms of enzymes, the NSPAs plus protease had a significant improvement on the AID of starch compared to control, as well as the NSPAs group. So, and this improvement is quite surprising because you can see in the control we got 97.6% starch digestibility, and in the combination, in the combo, we had 98.9%. So that was a surprising 1.3% increase in the ileal digestibility. For the protein sources, the canola meal diet had a significant higher starch digestibility compared to the soybean meal group. And we think this is probably something to do with the pectin content in the soybean meal. For the protein digestibility this time, unfortunately, we did not see any significant effects by the enzymes and protein sources, and there was no interaction. And we think this lack of effects by the enzymes is probably due to the endogenous secretion of proteins by the pigs themselves in the small intestine. So just a very short conclusion for this study, we found that the NSPAs plus protease combination had a significant improvement in the apparent ileal digestibility of starch in these ileal cannulated pigs. The canola meal diets also had significantly higher additive starch than soybean meal diets, and enzymes and protein sources did not show any effect on the protein digestibility. So by putting the 3 studies together, what do we know for now? The first thing is that we know these commercial enzymes, they are effective in improving the nutrient digestibility through both in vitro and in vivo studies in pigs at different ages, regardless of diets. The second thing that we know is that these enzymes, they have shown much more improvements in the nutrient digestibility in the post-weaning piglets, as what you have seen previously, than in these cannulated weaning green pigs, which are a bit older. And the third discovery was that the in vitro methods, now we know that they are very powerful tools for studying the mechanism of enzymes, and they are completely complementary to in vivo digestibility studies. What are the future prospects from the work in my PhD project? And these future prospects are mostly delivered from the limitations. The first limitation is that the in vivo digestibility studies that we conducted in the project, they did not capture the nutrient digestion kinetics or the rate of digestion. So that was the progression I was talking about earlier. And this digestion kinetics or digestion rate in the future can probably be done through several methods like catheterization to monitor the blood glucose change after each meal and to see if there are any enzyme effects. And the second limitation is that the digestibility studies by science, they are quite short, so they cannot really detect the accumulation of digestibility improvements into performance effects. So in the future, if there's any possibility, these enzymes should be studied for their effects on the pig's performance, preferably in commercial environment. So that's all for me today. Before I finish, I would like to acknowledge my advisory team and the funding bodies of this project for supporting my PhD research. And that's all. Thank you very much for listening.
Thank you so much, Xianyi. That was a great talk and really interesting data and prospects in there. So everyone, now's the time to think of your questions. Please put them in the Q&A tab and we will work through them. Can I just add one thing, Xianyi? Maybe we should acknowledge the University of Melbourne here, because that is with the location where the ileal cannulation experiment was done. They are the experts in this country on ileal cannulation in pigs, and it was a great collaboration with the group down there that enabled this study to take place. So thank you to them.
Thank you.
So yeah, there is one question there already from Eugenie. He says, great presentation, thank you, excellent results, particularly in protein digestibility. So he says, it's understood that low protein digestibility is one of the main causes of post-weaning diarrhea. So you observed a change in protein digestibility with the enzymes. Was there any noticeable change in post-weaning diarrhea that might be associated with the difference in protein digestibility caused by enzymes?
In our study, we had very low diarrhea rate among the pigs, and we believe that the cause of the diarrhea was due to lack of performance, more genetically rather than due to the enzyme treatment. So my answer to that question is we did not really see any effects on the enzymes, by the enzymes on the diarrhea rate.
But it was low in all cases, so it was
Yeah.
not something that was easy to pick up, I guess. Okay. Here's another question now from Anna. So she says one of the factors that govern the role of enzymes is particularly NSPases, is the solubility of the NSPs. So did you try to characterize the type of NSP, the soluble versus insoluble? So yeah, that's the first part. Let's answer that one first, and we'll come to a second part.
Yeah, so for the solubility of the non-starch polysaccharides, I do think that would be the next step to move on. And again, for the last study, since there's not enough time for me within my PhD, I did not conduct every analysis for now, but we still have the samples, and we will keep that comment as a direction for our next movement.
And the supplementary question of that is, should exogenous enzyme inclusion be fixed in pig diets, or should it be dynamic? I guess dynamic in terms of the age of the pigs, as well as from feed to feed.
Yes. So for me, I guess that's where the in vitro model kicks in, because we use the in vitro model to predict what can happen in vivo in pigs. And before you actually conduct trials, you just need to do some very simple in vitro experiments, and you can definitely see how the dosage and the enzyme type could work, not just based on the pig's age, also as the diet composition.
So maybe you could expand on that a bit. So reflect a bit on the where you do and do not see good correlation between in vitro and in vivo results. Could you just maybe give us a broad outline of how that's come through in your studies?
So if I may, I will go back to the in vitro results that we had. So a big limitation for now that we had with the in vitro model is, sorry, I'll just try to find that slide. I'll use the result on this slide here. So you can see pretty much for all the individual grains, when we did the in vitro digestion, the digestibility of those grains only reached 70 to 80%. And that is actually a major limitation that we had for now. We cannot reach the same numerical digestibility as what we have observed in vivo. And that's why I made the conclusion there that in vitro was a good tool to study the mechanisms of enzymes rather than to replace in vivo experiments. And regarding the correlation between the in vitro and the in vivo, in my study, I do think that there is a very good correlation. You see good effects by the enzymes, especially by the xylanase gluconase. And in the next slide, you see the xylanase gluconase here. They also improve the digestion rate and the amount of starch digested. Keep this result in mind if I go to the post-weaning trial that we did. The effect of xylanase gluconase can be seen as early as in the middle jejunum, which is the upper part of small intestine. So for me, this significant improvement by the xylanase gluconase in the middle jejunum means that it has already facilitated the digestion rate, particularly for the starch. But again, a drawback of this study, we were not able to measure the digestion rate. And that's something we will try to do in the future. And I hope that answers the question.
Yes, I think this is a more of a discussion than a simple answer, I guess, because I guess what you're trying to say is that there's an opportunity to integrate in vitro and in vivo methods. They each have their pros and cons, of course. And I guess maybe you could also reflect on you talked about the ileal cannulation being the gold standard method for determining digestibility, basically as a single point measurement. How do you see that being changed into the future? What sort of what are the pros and cons of sticking with ileal digestibility through cannulation measurement?
Yeah, I think that also um, reflect on what we have done for the post-weaning digestibility. At here for the ileal digestibility, these are just one data point measurements. So what you are seeing here, these numbers are just how much starch has been digested by the end of ileum, but we don't know how fast starch has been digested. For example, in the NSPS group at here, you do not see a significant improvement in terms of starch digestibility, but I'm just guessing that in terms of rate, the starch digestibility would have been much higher in the middle jejunum or in the proximal jejunum. But for the ileal cannulation, unfortunately, we put a cannula at the end, and that's all what we can see for now. And reflecting on the future, I think that's also something I've discussed. I think we should do a performance trial to look at the enzyme effects. That early improvement in digestibility, especially for starch as well as for protein, could have done much more in terms of performance. So that's what I think.
Oh, thank you. Yes. Always an interesting question, the balance between in vitro and in vivo work and the role that each can play. Okay, give people just one more minute to think of any more questions they want to put in the Q&A. Anything else that people want to talk about? Doesn't look like it. Looks like you're off the hook, Xianyi.
Oh, thank you very much.
Okay, so maybe we can progress to the next, the final slides.
Sure.
Thank you. So yes, first of all, thank you so much, Xianyi. That was a great presentation and really nice answer session as well. Please come back, everyone, for the next episode in the Coffee Science Seminar Series in 2 weeks' time by Professor Christine Beveridge, the Director of the Centre for Horticultural Science. She's got an intriguing and quite long title I'll read out to you. Inspired by plant development research, designing networks and pipelines that can be used for prediction across a range of activities in agriculture. I can't get any broader than that. Maybe that means she doesn't know what she's going to talk about yet, but maybe she got a very clear idea. Tune in 2 weeks' time to find out. Okay, so with that, I think we're done for today. And thank you so much. Please visit the webpage, the QR code's there. And you can sign up to be there. You can sign up to be a regular recipient if you're not already for notices about the science seminars. And let's hope to see you again at a future edition of the Coffee Science Seminars. Thank you very much and goodbye.